Hello, very good evening to the students who have joined. So kindly can you let me know in the chat box if my voice is clear and my video is visible, and also if you can see the slides. Please write in the chat box if my voice is clear and you can see the PPT and my video also. Okay, thank you so much. So we will wait for some time, then we will start today's session. Okay, we will wait for 2 to 3 minutes and then we will start today's session. Meanwhile, if you cannot clearly hear my voice or you cannot see the PPT, please let me know in the chat box. Okay. Okay, I think we can start slowly. Meanwhile, when others join, so I will just add them on. So, very good evening to the students who have joined in the today's session. My name is Moshumi Dev and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So, this is the week 6 live session for the NPTEL course of Medicinal Chemistry. Today, we will solve 10 to 20 to 20 around 20 questions regarding your week 7 course content, and this type of question will help you to solve the weekly assignments and also to learn the course better. Okay, so we will discuss 20 MCQs. When I show you some question, try to answer it in the chat box. It does not matter if your answer is correct or incorrect, but you should try to answer the questions in the chat box. I will check it, and after that I will show you. After you try to answer the question, I will show you the correct answer. Okay, and that's how we will do this session. Okay. Okay, I think there are sufficient students, so we can just start today's session. So if somebody try to join, I will just add them on. Okay. So okay, so this is the week 6 live session. Today's topic is your week 7 course contents. Okay, so let's see what are the questions, and when I show you some question, please try to answer the question in the chat box. Okay, it does not matter if it is correct or wrong, but it is important for you to try to solve those questions, because that's how you will understand like what is your level of understanding of the course. Okay, so let's start. So I think the screen is clearly visible and my voice is audible, right? If you have any problem hearing my voice or seeing the PPT slides, you can write in the chat box. I will just check it out. Okay, so I think it is clear. So now, so the first question of the today's session is, what is meant by a lead compound in medicinal chemistry? Your options are: number A drug containing the element lead. Option B, a leading drug in the particular area of medicine. Option C, a compound that acts as a starting point for the drug design and development. And option D, a drug which is first to be prescribed for a particular ailment. So you have to identify what is the lead compound in the medicinal chemistry. So when you are studying medicinal chemistry, you will hear a lot this voice like it is a lead compound for a drug discovery process or a disease, or it is a hit compound. So you should know what is the basic differences of this terms and what is these terms are actually meant. Okay, so try to answer this question, what is meant by the lead compound in medicinal chemistry. try to answer it in the chat box. Whatever you are thinking should be the correct answer, and you should write it up. Okay, it does not matter if it is correct or wrong, but at least try to answer this. Your options are: number A drug containing the element lead; option B, a leading drug in particular area of medicine; option C, a compound that acts as a starting point for drug design and development; and option D, a drug which is first to be prescribed for a particular ailment. So, do we have any correct? Okay, do we have any correct answer? Okay, so everybody is thinking it is option C. Okay, let's check out. Yeah, so the correct answer, very good, it is number C, a compound that acts as a starting point for the drug design and development. So, let us see what is a lead compound. So, a lead compound, a compound demonstrating a property likely to be therapeutically useful. The level of activity and target selectivity are not crucial. It is used as a starting point for the drug design and development and found by the design, molecular modeling or NMR, or by screening compounds, natural or synthetic. Okay. Need to identify a suitable test in order to find a lead compound. An active principle, a compound that is isolated from natural extract and which is principally responsible for extract's pharmacological activity, often used as a lead compound. So, these are the introduction for the lead compound. Now, what is the drug discovery cycle? As you can see, this is the drug discovery cycle. So, the first is the compound collection. Okay. And this collected compounds are go to the primary assays, high throughputs and in vitro screenings. After that, we go to the secondary assays like counter screens, bioavailability, toxicity, metabolism, these things. And these two primary and secondary assay gives us the lead compound and the SAR. Okay, sometimes the secondary assay it itself goes to the as a clinical candidate. Okay, now what happened? Like this lead compound indirectly or directly goes to the structural characterization of the protein ligand complex and the designing, and then chemical synthesis happens, and this chemical synthesis gives us the compound which is again goes to the primary assay and then secondary assay and then goes to the clinical compound. So, first you have some compounds, you try it for the primary and secondary assays. From that you identify a lead compound and perform the structure activity relationships. After that the structural characterization and complex ligand formation takes place, and when it is completed the new compound or new novel compounds are designed. After the design the chemical synthesis are done, and after that what happened that newly synthesized compounds again must go through the primary and secondary assays, and after that we get our clinical candidate. So, I think it is clear what is a lead compound in medicinal chemistry. Okay. So, now I will move toward your next question. So, yeah, like every day I say, like if you have any doubt, any query at any point of any question, you can write in the chat box at the end of the session or in between. When I check the chat box, I will definitely try to answer your queries. Okay. And so, let us check if we have any doubt for this question. Okay. So, I think it is clear to everybody. Now, I will move toward the next problem. Okay. So, problem number 2, it says that which of the following drugs was not isolated from the natural source. So, your options are number A, quinine, number B, morphine, number C, artemisinin, and number D, isoniazid. So, you try to identify the drug which is not isolated from the natural source. See, natural products and natural source is a high, it is a very good source for the drugs, and like in previously many drugs are isolated from the natural source even today also, and you all know like very Indian traditional medicines and everything, it comes from a natural source, like in ancient Indian sources or drugs in Ayurveda, everything comes from a natural source. So, now you try to identify among these 4 which is not isolated from the natural source. Try to write the correct answer or whatever you are thinking to be right in the chat box. I will check it and then I will show you the answer and we will discuss this. So, do you have any correct answer for this? Okay, we have isoniazid C, D, C. Okay, I think we have two main options which you are thinking should be might be correct. Okay, let's check which is the correct answer. So the correct answer is number D, isoniazid. Okay, so let's check others. What are things? So this is the structure of the isoniazid. So it is a pyridine molecule which has this carbonyl carbon and this NH2 bond. So this isoniazid is a first line medication in prevention and treatment of tuberculosis. So it is synthesized in early 20th century and it is available in tablet, syrup or injectable forms given intramuscularly or intravenously. And isoniazid is manufactured from isonicotinic acid which is produced from 4-methyl pyridine. Okay. So now let's check the structures of the other. So this is the structure of quinine, this is the structure of morphine and this is the structure of artemisinin. So all of these compounds, this quinine, morphine and this artemisinin, these are isolated from the natural source. So as you are somebody was confused for this isoniazid and artemisinin. So artemisinin is also it is also taken from a natural source. So it is a traditional Chinese medicine which is used for curing malaria. Okay, so it is it actually has gotten a Nobel Prize in I think 2015. The person who discovered this natural product and successfully it is isolated and today it is widely used for the treatment of malaria. Okay, so this was all about this drug natural source. So I think it is clear to all. So let me check once if we have any doubt or anything. Okay, so again we will move toward our next problem. So problem number 3, it says that what is the term used for drugs that are similar in structure to a known drug and which are used for the same purpose? Option A, copycat drugs. Option B, me too drugs. Option C, derivative drugs. And option D, analog drugs. You have to identify the term which is used for the drugs that are similar in structure to a known drug and which are used for same purpose. So is it copycat drug, me too drugs, derivative drugs or analog drugs? Try to identify the correct option in the chat box. Okay, let's check if we have any correct answers or any answers for it. Okay, we have two answers. Okay, we have many answers I think. Okay, so me too drugs, option D, option B, option B. Okay, let's see what is the correct answer. So yeah, this is good, like everybody is trying to answer because it does not matter if the answer is correct or wrong, but it is very important to interact in this type of class. Okay, so let's check. Yeah, so very good. So the correct answer is number B, me too drugs. So what is me too drugs? So the term me too drug or follow, okay, once again, let me check. So the term me too drug or follow on drug refers to a medication that is similar to a pre-existing drug, usually by making minor modifications to the prototype reflected in slight changes in the profiles or side effects or activity and used to treat conditions for which the drugs already exist. Me too drugs include diazepam, ranitidine and esketamine. So these are the examples of the me too drugs. Okay. So the definition, a drug that is structurally very similar to already known drug but with only minor differences. The term me too carries a negative connotation. However, me too products may create competition and drive prices down, and also it is known as follow on drugs. So, me too drugs can be broadly defined as chemically related prototype or other chemical compound which is an identical mechanism of action. Okay. So, if you this question it is if given as a me too drug, so this might also be given as the follow on drug. So, it is very important for you to remember the other terms for the same thing, right. See here the question I am given as the me too drug. So, the question may also be given in the form of the follow on drug, this term. So, you try to remember like this me too drug and follow on drug refers to the same thing, and what is this thing, and this is the and the examples of the me too drugs, like these are the 2 to 3 examples. So, try to remember these examples, because the question can come like this, you are given some names of the drugs and you need to identify which kind of drug it is. Okay. So, this was these are the some examples like diazepam, ranitidine and esketamine. So, these are the examples of the me too drug or follow on drug. So, I think it is clear. I will now move toward the next problem. So, problem number 4, it says that there are several sources and methods of discovering new compounds. Which of the following is most likely to lead to the discovery of the complex structure quite unlike any other previously discovered? Number A, combinational, so there is a printing mistake, combinatorial chemistry, so it should be combinational. Okay. So, there is some printing mistake here. Number B, database mining. Number C, screening plant extracts, and number D, me too drugs. So, as we all know, in drug discovery process there are several sources and methods for discovering new compounds. So, which of the following is most likely to lead discovery of a complex structure quite unlike any other previously discovered? So, there is a spelling mistake here, it should be combinational chemistry, then database mining, screening plant extract and me too drugs. Right, so, try to answer this question, try to figure out what should be the correct answer and write in the chat box. Let's see. Okay, so option A is the most cell. Okay, let's check if the option A is correct or not. Okay, so the correct answer for it is the screening plant extract. See, combinational chemistry, it is something which is we will discuss later on, but since the question, if you read it carefully, it says that, okay, see. So, the one thing you should clearly look at is in the question, it says that the discovery of a complex structure. So, as we all know, this the complex structure, the natural products actually have the most complex structure. Okay, synthesized drugs are fine, synthesized structure, synthesized things, these are fine, but the natural product, the complexity of the natural products is like anything else. Okay. So, if you check the previous answer or something we discussed earlier, yeah, this one. So, as you can see, like this quinine, this morphine or this artemisinin. So, can you see the structure? So, the structures are so much complex. Okay. Now, there are so many chiral centers, so many particular bonding, so many complexity. So, this natural, always try to remember, like synthesized drugs are fine, but natural products have the most complexity, because the nature is the best or the brilliant chemist, should I say. So, as you can see, like this is the approach of the taking the compound from a plant. Okay. So, the ethnopharmacological approach, random approach or approach based on the traditional system of medicine. So, it gives to the plant section. So, the plant section, after it goes to the authentication. So, what happened in this authentication, the taxonomic, macroscopic, microscopic, chromatographic and spectroscopic techniques have been used, and then chemometric immunoassays and DNA fingerprinting. So, after the extraction, this plant extract we have. So this plant extracts goes from the fractionation and the isolation and biological screening, and this biological and toxicological screening takes place. After that we get this pure compound. So from the pure compound we go to the structural elucidation and the synthesis of that compound particular, and then it goes to the biological studies and toxicology, and goes to molecular modeling or in silico screening, structural modification for the new analogues. So what happened, first we discover a new compound or a new lead from the plant extract, and then after the extraction of we see the structural basis and visualize it by the molecular modeling and computational methods like in silico screening, and then structural modification gives us the new analogues. That pure compound also goes to the biological screening for the toxicology and other characteristics. So that this is the about the plant extracts. Okay. So is it clear to all? Because there were some, so there were some I think difficulty for this question. So always try to remember, like this natural products have the most complex structure unlike any other thing. Okay, and when you are studying something, so always try to remember, try to read the question carefully. Okay, so this was your problem number 4. Now we will go toward the problem number 5. So the problem number 5, it says that which of the following needs to be established before the search for a new lead compound takes place. Number A, the pharmacophore. Number B, structure activity relationship. Number C, a bioassay. And number D, patents. So you have to identify which of the following needs to be established before the search for a lead compound takes place. Is it pharmacophore, structure activity relationship, a bioassay, patents? What is it? Try to answer this in the chat box. It does not matter if it is correct or wrong, but you should try to answer this. Okay, so we have some answers, mainly C and bioassay. Okay, let's check. Okay, so very good, it is the bioassay. So what is a bioassay and what is the principle and applications of bioassay? So the basic principle of bioassay is to compare the test substance with the international standard preparation of the same and to find out how much test substance is required to produce the same biological effect as produced by the standard. And the application are according to the several pharmacospace assay used for estimate the potency of the natural drug for which chemical method is not known or established, standardization of drugs of a natural origin, plant or animal origin whose structural origin is the unpredictable, screening of new compound for biological activity, estimation of biologically active substance like histamine, acetylcholine, 5-hydroxy, tryptamine, adrenaline, bradykinin, substance P, prostaglandins, estimation of ED by ED50 by TD50 and LD50. Presently instead of LD50, NOEL and LD10 are preferred. Okay, so what is this term ED50 by TD50 stand for? If I remember, I discussed it in last class or in previous. So this is a particular term which is used for this ED50 by TD50. So if you know, you can write in the chat box. If you does not remember, I will just tell you again. So this is about the bioassay. So the other terms, which is pharmacophore. So pharmacophore is the active part of the drug structure. So in the drug structure there is something which is very much novel and you should not change the structure. Keeping that part intact, we actually vary the other substituent to check the activity. Okay, so that fixed part is called the pharmacophore, and structure activity relationship is that same, like we change the other substituent to see how the activity changes. And patents is something very different thing. So we will actually discuss this thing later on. So did you understood this part? So good. So yeah, as I said, like this ED50 and TD50 by TD50, it is known as the therapeutic index. Okay, so let's go to the problem number 6. So it says that which of the following is not an endogenous compound? Option A, dopamine. Option B, endorphin. Option C, noradrenaline, and option D, desipramine. So identify which of the following is not an endogenous compound and try to answer it in the chat box. So your options are dopamine, endorphins, noradrenaline, and desipramine. So which of the following is not an endogenous compound? Take 1 or 2 minutes and try to answer this in the chat box. Okay, so we have, I think, few answers for it. Number 6, D. D and C. We have two answers, D and C. So let's see which is the correct answer. Yeah, so the correct answer is number D, desipramine. So this is the structure of the desipramine, and all the others like dopamine, endorphin and noradrenaline, these are the endogenous compound. Okay, and this is the desipramine hydrochloride, it is given as a form of the tablets. So what is the endogenous lead compound? So let's see a little bit about it. So endogenous lead compounds often are often simple and flexible. Okay, like adrenaline. Adrenaline also endogenous lead compound. So they fit several targets due to the different active conformation, like androgenic, adrenergic receptor types and subtypes. So there is a flexible chain, and based on that flexible side chain, okay, the rotation happens and the different conformation can be taken up from this endogenous compound. So rigidify molecule to limit conformational restraint. So increases activity, more chance of desired active conformation, increases selectivity, less chance for undesired active conformation. And the disadvantages is the molecule more complex, may be difficult to synthesize. So this is a thing called rigidification. Okay, and endogenous compounds are often flexible and simple. To make it more rigid, this thing can take place. So as you can see, like this desipramine it is not an endogenous compound. So as you can see, there is a cycle a chain, there is no flexible chain between this two phenyl rings. Okay, it is connected by a connection, and that is why it is very rigid and it is not a endogenous compound. So now we will move toward the problem number 7. So problem number 7, it says that what is the term used for an animal that has been genetically modified for the in vivo test? Your options are number A, chimeric animal, number B, transformed animal, number C, hybrid animal, and number D, transgenic animal. So identify the term used for an animal that has been genetically modified for the in vivo test. Is it chimeric animal or transformed animal, hybrid animal or transgenic animal? Try to answer it in the chat box. So let us check if there are any answer for this question. Okay, so D and C, again we have two different answers for it, D and C. Okay, no problem, let us check it. Okay, so the correct answer is number D, transgenic animal. So this is the term which is used for an animal that has been genetically modified for in vivo test. So what is the definition? So the definition of this transgenic animal is that an individual in which a gene or genes were introduced by one or other technique of transfection to a genome of an animal is called the transgenic animals, or a transgenic animal is one that carries a foreign gene that has been deliberately inserted into its genome. Okay. So the gene introduced by the transfection is called the transgene, and the transgenesis may be defined as the introduction of exogenous DNA into the genome such that it is stably maintained in a heritable manner. Okay. So this was the definition for this transgenic animal. So what is a transgenic animal? So there is an animal where the genes or the different hybrid genes are introduced by a external method. Okay. And what is the difference between this chimeric and transgenic organism? So the chimeric organism, so first this is the definition. Chimeric organism is a single organism composed of cells with more than one distinct genotype, and transgenic organism is an organism created combining of DNA from different genomes or the insertion of foreign DNA into the genome from one organism to another. The process, the first one, a result of combining the cells of two or more genetically distinct individuals, and the second one, a result of combining foreign DNA into an organism. So the first one, changes to phenotype. So it does not always bring out changes to the phenotype of the individual, and the second one always brings out changes to the phenotype of the individual. In a new individual have cells with more than one distinct genotype for the chimeric organism, and for the transgenic organism new individuals have transgene. Now the example, the first one is the chimeric mouse, and the transgenic organism is the transgenic plant, animals or microorganism. So this was the about the chimeric organism, transgenic organism, the differences between these two, and this is all about the transgenic animal. Okay, so remember this thing, it is a very important term you should remember for the drug discovery purpose. So I think it is clear. So do you have any doubt regarding this question or any question previously discussed? So if you have any doubt in between the session, you can just tell me the doubt if I have to repeat anything, or you do not understand some points. So just tell me that time I will just try to answer these questions, okay, when you have any doubt. So till now, do you have any questions regarding any problem or any doubt? Do I need to repeat any question, any part? Okay, next. Okay, so I will move toward the next problem. Okay, so problem number 8, it says that which of the following is the correct definition of bioavailability? Number A, bioavailability describes the proportion of the drug administered that is metabolized very quickly and thus is not available to induce a physiological effect. Number B, bioavailability describes the ability of administered drug metabolites to cause undesirable physiological effect. Number C, bioavailability is used to describe the fraction of the dose of a drug administered that is present within the body and facilitates the desired physiological effect. And number D, bioavailability is the length of time an administered drug is present in the body and thus is available to cause physiological effect. So, you have to identify the correct definition for the term bioavailability. So, read every point, every option very carefully and try to understand and try to answer the correct definition for the bioavailability. So, again I will repeat the question, which of the following is the correct definition of bioavailability? Number A, bioavailability describes the proportion of the drug administered that is metabolized very quickly and thus is not available to induce physiological effect. Number B, bioavailability describes the ability of administered drug metabolites to cause undesirable physiological effect. Number C, bioavailability is used to describe the fraction of the dose of the drug administered that is present in the body and facilitates the desired physiological effect. And number D, bioavailability is the length of time an administered drug is present in the body and that is available to cause physiological effect. I think somebody wants to join. Okay, so we have two answers till now, C and D. So, again try to read it carefully and take 2 to 3 minutes to read it again, and others also try to answer this question, what you think to be the correct answer. Okay, let me show you the, so do we have any other answers for it? C, 2C and 1D we have till now. Okay, let us check which is the correct answer and understand what is the bioavailability. Okay, so the correct answer is number C. Bioavailability is used to describe the fraction of the dose of drug administered that is present within the body and facilitates the desired physiological effect. So, as I said, bioavailability is the fraction of administered drug that reaches to the systemic circulation, and it is expressed as a fraction of administered drug that gains access to systematic circulation in a chemically unchanged form. So, for example, if 100 mg of a drug is administered orally and 70 mg of this drug is absorbed unchanged, then the bioavailability is 0.7 or 70%. Okay. So, the definition, fraction of the drug administered reaching to the reaching systematic circulation, significance, dosage of drugs with a high hepatic extraction. So, the drug in tablets. So, I think this flowchart is not clear much, but I will when I will share this PPT with you, you can just check directly from the PPT and then you will just answer. I will just read it out. So, the drug in the tablet, it is a dissolved drug and undissolved drug. Okay. And the contents in this it reaches to the stomach and intestine, and there it is dissolved and something it is non-dissolved. Okay, at part it is non-dissolved. Then it goes to the gut wall where the drug is absorbed, and some fraction of the drug is absorbed and some fraction is not absorbed at all from the gut wall. Then the absorbed part, it goes to the liver. Okay, and from the liver it goes to the portal vein, and the loss during the liver to the gut liver passage. Okay, and the drug which is reaching to the portal vein, it is the fraction of the drug which is the bioavailability. and the bioavailability loss is the 1 minus F. Okay. So, as we saw here, if the 100 mg drug is administered to your body, so first what will happen if it is given I mean in a tablet form. Okay. So, let us say the drug is given in a tablet form, and in the 100 mg tablet you are taking with water in the mouth. So, first the drug will reach to the stomach, right. So, in the stomach we have acid and digestive liquids, the digestive fluids, and the fluids what will they do, they will dissolve the drug. So, after the drug is dissolved, so it will not dissolve completely. So, some part will be remain undissolved and some part of the drug will be dissolved. Okay. So, what will happen once it is dissolved, the dissolved drug it goes to the gut wall. Okay. In the gut wall it will be absorbed. The part which is absorbed, and again not the completely which is dissolved drug goes to the gut wall, it gets absorbed from the gut wall. Okay. So, some part will be absorbed, some part will not be absorbed. So, that absorbed drug what will happen, again it goes to the liver, and from the liver it goes to the systematic circulation or in the blood circulation. Okay. And during this passage from gut wall to liver, what happened, some fraction is also getting loose. So, the final fraction of the drug, so say you are taking 100 mg of the drug, in stomach you are losing 10 mg, in the gut wall you are losing again 10 mg, and in the during the passage from the gut wall to liver you again lose 10 mg, say it is an example. Okay. I am not saying it will go do like it. So, for example, it gives so 30 mg you are losing from that part. Okay. And the fraction which is reaching to your blood, it is 70 mg. So, what is the bioavailability for the particular drug? So, it is 70 mg divided by 100 mg means 0.7 or 70%. Okay, it is the bioavailability. So, for a drug it is very important for to make it a very the bioavailability should be very good. Okay. Otherwise, if it is it has poor bioavailability, what will happen, you are taking so much amount of drug, but it is not reaching your systematic fluids. Okay. So, which is not good for a particular drug. So, when you are designing some drug, so the characteristic of bioavailability is very important. Okay. So, I hope you understood this part, what is bioavailability, how we describe the bioavailability. So, do we have any doubt for it? Okay. So, as I say, whenever you are having any doubt regarding any question, so try to write it up in that moment also, because I will try to answer it as early as possible or at the end of the session, but when you are having some doubt, you just write quickly in the chat box. Okay. So, this was the problem number 8. Now, I will move toward the problem number 9. Okay. So, which of the following reflects the order in which various stages of drug discovery and development takes place? Number A, determining a target, establishing bioassay, finding lead compound and structure activity relationships. Number B, establishing a bioassay, determining target, finding lead compound and structure activity relationship. Number C, determining a target, establishing a bioassay, structure activity relationship, finding the lead compound. Number D, determining the target, structure activity relationship, finding the lead compound, establishing a bioassay. So, now you have to identify which is the correct order for the drug discovery and development process. Read all the options carefully. Think about the thing we discussed so far in today's class and on the previous classes also, regarding target, regarding bioassay, lead compound, SAR, everything. So, take everything in count and then try to answer this question. First, read the question and the answers very carefully. Take 2 minutes and read the question answers very carefully, and then try to identify which is the correct order in which various stages of drug discovery and development takes place. Do we have any answers for this question? Okay, so we have D, then again D, C, A. Okay, so we have a different variety of answers for this question, which is good because everybody is thinking in different manner. Okay, so let's check the correct answer. So correct answer is number A. So it is determining the target, establishing the bioassay, finding the lead compound and structure activity relationship. So as I discussed, I think in a previous one question, like first you have to establish a bioassay for finding the lead compound, and as we all know, before finding the lead compound you cannot perform the structure activity relationship, but all those things will be done after we determine our perfect target. Okay. So, target determination should be the first point for the drug discovery and development process, because if you do not know what is the target, what is the disease you want to work on, you cannot start working, right. So, it is the first stage for the drug discovery process, like determining the target. So, once you determine the target, you have to establish an assay, a tool, and then we have to find the lead compound and structure activity relationship. So, understanding the drug discovery process, the process of discovering, testing and eventually gaining approval for selling a drug is a long and arduous one. Here we look at the different stages involved and approximate the length of the time that each stage takes to eventually arrive at an approved drug that can be given to patients. Okay, so it is a very as we say, it is a very long term process. So, basically a drug discovery process, a drug to come in the market, it takes nearly 10 to 15 years. Okay. So, 10 to 15 years it is the time which it takes for the drug to be approved for the use of the patient. So, the first stage is the research and development phase. Okay, and this research and development phase, it takes nearly 3 to 6 years. Okay. And what happens in this research and development phase? So, drug development begins long before the clinical testing. It starts with the identification of the target for a drug to action, then identification of the compounds that could be potentially hit the target. So, the first step is the target identification. So, what will be the drug effect? Understanding of the causes of the disease or condition can help the researchers to know what processes or pathways drugs to treat the condition need to be able to target, which is the and then it is the compound screening. So, plus 10,000 plus compounds are screened in laboratory tests for their ability to affect the identified target. They are also screened to check they do not interfere with other related targets, which is very important, because when you are given a particular drug to your body for a particular disease or target, it should not hamper other processes or other targets. Okay. And the lead identification, which compounds to test through the screening is likely to uncover a perfect drug candidate. Promising candidates can be identified. The structures of these molecules can be modified to try and improve their activity. So, this is the first step of the research and development, which takes nearly 3 to 6 years, and it is the most laborious stage. Okay. Then it goes to the preclinical studies. So, it takes nearly 1 year for the preclinical. So, up to 10,000 compounds considered during the screening, but only around 250 will make it to the preclinical testing. Efficacy and potential risks are evaluated before human trials can begin. In vitro studies, Latin, the in vitro means it is a Latin word for the in glass. So, laboratory experiments on cells or molecules outside their usual biological surroundings. This gives some idea of the efficacy against the identified target. Toxicity can also be assessed. In vivo studies. So, in vivo is the Latin for the in the living. So, drugs that look promising in the in vitro testing will process the testing in the animals. So, these include tested on toxicology, efficacy and how quickly the drugs are absorbed and subsequently eliminated. Okay. And then required standard minimum 2 mammalian species. Drugs must undergo toxicity testing on at least 2 mammals, 1 non-rodent, including at least 2 administered route before they are allowed to process to the clinical trials in human. So, this is the preclinical study, which is the second stage. Now, The third stage, it is the clinical trials, which takes again a very laborious time for 4 to 7 years. So it is the clinical trials involve the human patients. This test in the volunteers provide information of safety and efficacy. Around 70% of the drugs succeed in phase 1, 33% phase 2 and 25 to 30% in phase 3. So phase 1 trials usually 20 to 80 people. So the primary goal of phase 1 trials to determine the drug side effects. Additionally, how quickly the drug is metabolized and excreted from the body can be determined during these trials. Phase 2 usually done on 100 to 300 people. So phase 2 trials help to determine how effective the drugs in the patient who have condition it is trying to treat. Controlled trials compare the effects of the drug to that of the placebo. And phase 3 trials usually 1000 to 3000 people. So gauges efficacy, dosage and safety in a larger population. Also compares efficacy of existing treatment as well as interaction with other drugs and effects of the different doses. So it is the phase 3 of the drug discovery process. Now the phase 4 is the review and approval part. So it takes nearly 1 to 2 years. So if a drug is deemed to effective in clinical trial, it is submitted to the regulating bodies to be approved. So it is estimated that only 1 in 5000 drug candidate makes it all way to approval. Evaluation evidence from the trial. The regulatory body reviews the evidence provided by the pharmaceutical company for the drug's efficacy and safety. Drugs offering important advances are given priority. Approval and manufacture, approximately 75% submission are approved. So it is if the benefits of taking the drug are deemed to outweigh at risk, it is approved, and the cost of developing a drug gains marketing approval, estimating $2,558 million. And post release monitoring, it is the indefinite duration it takes, okay, for the post release monitoring. After a drug is approved and available for use by the patient, it is still monitored for any side effect in the general population that may not have occurred during the drug trials. So it is all about the drug discovery process. So as you can see, it is a very tedious and laborious job, but also very rewarding. So this is all about the drug discovery. So I think it is clear to all. So do we have any doubts for this question? Okay. So now we will move toward the problem number 10. So problem number 10, it says that which of the following is not classed as an in vivo test? Number A, testing anti-cancer drugs on a tumor grown on the back of a rat. Number B, testing analgesics by determining a guinea pig sensitivity to pain. Number C, testing antibacterial by measuring the growth of the microorganism on a petri dish in their presence. Option D, testing antibacterial agents in the infected rats. So previous in the last question we discussed a little bit about the in vivo testing. Now you try to understand what is in vivo testing. We will discuss it in a very detailed manner, but try to answer this question. So which of the following you think is not classed as an in vivo test? Number A, testing anti-cancer drugs on a tumor grown on the back of the rat. Option B, testing analgesics by determining a guinea pig sensitivity to pain. Number C, testing antibacterial by measuring the growth of the microorganism on a petri dish in their presence. And number D, testing antibacterial agents in infected rats. So try to identify which of the following should not be classed in the in vivo testings. So, do we have any correct answer for this? So, the number C. Okay. So, the number C is the only answer we have got for this. Okay, let's check. So, yeah, very good. It is the number C, the testing antibacterial by measuring the growth of the microorganism on a petri dish in their presence. So, it is a in vivo test, in vitro test. Okay. So, let us check out what is the differences between the in vivo and in vitro. So, in vivo testing system. In vivo testing plays a vital role in the drug discovery involving animal testing and clinical trials. So, before a drug candidate is tested in the human subject, it must undergo animal testing to further evaluate the toxicity, metabolism over efficacy. Vertebrate animals models such as primates, dogs, rabbits are most commonly the preclinical study. So, it is the, as you can see, when the compound is in the glassware or any laboratory system. So, it is the in vitro testing, then it goes to the ex vivo system and in the in vivo system, and it is a preclinical study before goes to the clinical study. So, what is the difference between in vitro and in vivo? In vitro refers to a phenomenon in which a given procedure is performed in a controlled environment outside the living organism. In vitro refers to a phenomenon in which experiments are performed using a whole and living organism. Dead organism or isolated cellular components are used for the in vitro testing. A whole living organism is used for the in vivo testing. In vitro, it is performed under controlled laboratory conditions, and in vivo, it is performed under physiological conditions. The first one is less expensive, the second one is expensive. For the in vitro, it is less time consuming, and the in vivo, it is more time consuming. The in vitro, it is less precise, and in vivo, it is more precise. For example, cell culture experiments in petri dishes and experiments in the test tubes are the in vitro testing. Drug testing experiments performed by using model organisms such as mice, rabbit, apes, it is the in vivo example of the in vivo testing. In vitro fertilization refers to artificial fertilization method in which the fusion of male and female gametes occur outside the human body. Regular fertilization mechanism in which the fusion of male and female gametes occur within the body, it knows as the in vivo fertilization. So, it is all about your in vivo and in vitro method. Okay. So, I think it is clear what is in vivo, what is in vitro. Okay, now I will move toward the next problem. So, as I show you the problem number 11, try to solve it. I will give you a 5-minute break, not 5-minute break, I will give you 2 to 3 minute break. Okay. So, read the problem number 11 carefully, try to answer it. I will be just here, my camera and sound will be off for 2 to 3 minutes, and you also can take a break. So, before going to the break, let us see what is the problem number 11. Okay, so the problem number 11, it says that in which of the following process the protein 3D structure of the target is must requirement. Number A, ligand based drug discovery or LBDD. Number B, structure based drug discovery or SBDD. Number C, fragment based drug discovery or FBDD. Number D, all of the above. So, you have to identify in which drug discovery process the protein 3D structure of the target is a must requirement. Okay, so you try to answer this question, take 2 to 3 minutes to answer this question. It is a break also. After answering the question in the chat box, you can take a break also. So, my camera and sound will be off for 2 to 3 minutes, but I am here. So, do not leave the the class. Okay. So, try to answer this question. After I come back in 2 minutes, we will discuss it. Okay. So, yeah. Okay, so take 2 to 3 minute break. In while you are taking the break, try to answer the question number 11. Okay, after coming back in 2 minutes, I will discuss the question number 11. Okay. Hello. So is my voice audible and my video is visible? So the break is over. I hope you guys are on the call. Okay, so for the problem number 11, we have I think few answers, number D and A. So let's check what is the correct answer for the problem number 11. Okay, so the problem number 11 was, in which of the following process the protein 3D structure of the target is a must requirement? Number A, ligand based drug discovery. Number B, structure based drug discovery. Number C, fragment based drug discovery. And number D, all of the above. So let's check the correct answer. Okay, so the correct answer is the structure based drug discovery. So for the structure based drug discovery process, the information regarding the protein's 3D structure of the target is a must requirement. Okay. So now we will discuss it in more clearly. Okay, thank you. Like my voice is audible. Okay, so what are the different methods for the drug discovery? So this is a very little different kind of question. Okay, so now till now we were discussing the question from your previous year of assignments, but now we will have 10 question which is not out of your assignment part. So try to answer those questions also, because it is related to the course content, but it is not directly from the previous year assignment. Okay. So the de novo drug design. Okay, let me use it. Okay. So there are different type of drug design. There is structure based drug design, ligand based drug design, atom based drug design and fragment based drug design. So for the structure based drug design, the receptor 3D structure is known and it is required. Okay. Generation of interaction sites for the ligands. It is happened in the structure based drug design. So in the ligand based drug design, the receptor 3D structure is unknown. So you don't know the receptor 3D structure. The generation of a pseudo receptor or direct similarity search takes place here. For the atom based drug discovery, a seed atom is required, high number of generated structures with the questionable chemical accessibility. And for the fragment based, a seed fragment is required, lower the number of generated structure with the good chemical accessibility. Okay. So for the others we will discuss later on the today's session or other sessions also, because we will come to this parts then later on. So today we will discuss only for the structure based drug design. So it is the workflow of the structure based drug design project. So first what happened? The crystal structure of a protein available from the literature. Okay, and the let's say you are targeting a particular disease, and you from the literature you just try to figure out what are the protein which is involved in that particular disease, and which protein you can target for your drug discovery. Okay, once you target the protein, you have to just see the crystal structure for the protein which is available in the protein data bank. Okay, so there is a website called protein data bank. There we will get the crystallized protein structure, and you have to download the protein from in a format which is called the PDB format. Okay, protein data bank format. So all the proteins which are present in the in the human body, not all the proteins, or all the protein which are characterized till now, all the information you will get from the protein data bank. So once the crystal structure of the protein it is available from literature, and you identify the crystal 3D structure, now what we go to we will go to the high throughput virtual screening, and in this virtual screening you will identify some hit compounds. Okay, so the hit compounds are the compounds which are able to interact with the your protein 3D structure or your target. And from this identification of the hit, you have what you have to do, you have to synthesize or purchase the hits. Okay, so the hit compound can be readily available, so you can purchase it from the any chemical website, or you can synthesize the compound if you don't have the direct availability of the hit. And after identification of the hit, what happen, we have to perform the biological assays, okay, or the normal assays to identify the active compounds of the molecule, and from there we will go to the directly to the lead compound, and we have to optimize the lead to synthesize further compounds. Okay, so it is a basic workflow of the SBDD or structure based drug discovery process. Okay, so I think it is clear. So it is something different thing. So remember, for the protein 3D structure of the target is must requirement for the structure based drug discovery. So now I will move toward the next problem. So problem number 20, it says that identify the correct statement about the combinational chemistry. So in previous one question there was something in combinational chemistry regarding, I have said I will discuss this later. So identify the correct statement about the combinational chemistry. Number A, one molecule can be done at one time. Number B, it has a high risk of failure. Number C, it was developed using solution phase synthesis. Number D, it creates an impact in the lead discovery and optimization. So try to answer this question. It does not matter if your answer is right or wrong, but at least read the question carefully, read the all options carefully, and pick out the option you think might be correct about this question. Eventually I will discuss, but you know, in any examination or anything, you will not get the question which is completely known to you. Sometimes you have to guess the answer, and it is also a very important technique to learn. Okay, so read the options carefully and try to identify which is the correct statement regarding the combinational chemistry. So the options are: number A, one molecule can be done at one time; number B, it has high risk of failure; number C, it was developed using solution phase synthesis; number D, it creates an impact in the lead discovery and optimization. So we have any correct answer or any answer for this? So I think number D. So I don't know number D is for which question. Okay, let's check out. Okay, so the correct statement regarding the combinational chemistry is, yeah, number D, it creates an impact in lead discovery and optimization. So as you discussed like thing, I told you like read the options carefully, and I think number D is the most likely to be the correct answer for this. Okay, so it's fine. Let's discuss about what is the combinational chemistry. So for the definition, combinational chemistry is a technique by which large numbers of different but structurally similar molecules are produced rapidly and submitted for the pharmacological assay. This technique uses the same reaction condition with the same reaction vessels to produce a large range of analogues. Technique invented in late 1980s and early 1990s to enable tasks to be applied to many molecules simultaneously. And what is the combinatorial library? The okay, I think there is a printing mistake. It should be combinatorial. So it is, there is a printing mistake here. So I will just write it later. So combinatorial should be the correct pronunciation and the spelling. So I will just correct it later on. So what is the library? The products produced from the combinatorial synthesis are known as the combinatorial library. Libraries may be a collection of the individual compounds or mixture of compounds. Screening the compounds of a library for the activity using high throughput screening techniques enables the development of the development team to select the suitable compounds. So this is the flow chart. So first what happened, like target therapeutic target is identified, then the lead discovery happens, then we go to the lead optimization, development candidate, and the drug. So I have told you about this flow chart in the previous time also. So the combinatorial chemistry, it actually affects this part 1 and this phase 2 and phase 3 in the lead discovery and the lead optimization. Okay. So what is the difference between the conventional and combinatorial chemistry? So in the conventional, one molecule can be done at one time. Okay, but in the combinatorials, many molecules can be done at one time. So in the conventional, first we have to make, then we have to purify, and then we have to test. So make, purity, and test. But for the combinatorial, first make, then test, and then purify. Okay. The hundreds of molecules can be done for the conventional chemistry, but from the combinatorial chemistry, thousands of molecules can be done in 1 month. So in the first one, slower lead generation; in the second one, faster lead generation. The conventional, it has a high risk of failure, but the combinatorial, it has low risk of failure, and this leads to the synergy and the lead identification. Okay, so there is some printing mistake. I will just I will just write it right now. So I think it is clear. I will then move toward the next problem. Okay, so read this question carefully. Problem number 13, it says that pores in the nephron cell membrane which are specific for the reabsorption of water molecules and prevent the passage of the ions and other molecules are called number A glomerulus, number B arterioles, number C aquaporins, number D none of this. Okay, I thought there was somebody wants to join. Okay, so again I will repeat the problem number 13. Pores in the nephron cell membrane which are specific for reabsorption of the water molecules and prevent the passage of ions and other molecules are called number A glomerulus, number B arterioles, number C aquaporins, number D none of this. So try to identify the correct option. Okay, so do we have any answers for this question? Aquaporins A, C, A, okay, good. Okay, so the correct answer is number C aquaporins. Okay, let me hold that, there is a pointer. Okay, so what is aquaporins? So in the nephron it is surrounded by the rich network of blood vessels that carrying the filtered blood away from the glomerulus, permitting much of the contents of the nephron to be reabsorbed into the blood supply. Most of the water that was filtered into the nephron is quickly reabsorbed through the pores in the nephron cell membrane, which are specific for the water molecules and bar the passage of ion or other molecules. And these pores are made up of the protein molecules which are called the aquaporins. Aquaporins are also called the water channels. They are the integral membrane proteins from a larger family of major intrinsic protein that from the pores in the membrane or biological cells, mainly facilitating the transport of water between the cells. The cell membranes of a variety of different bacteria, fungi, animal and plant cells contain aquaporins through which water can flow more rapidly into and out of the cell than the diffusing throughout the phospholipid bilayer. 6 membranes spanning alpha helical domain with both carboxylic and amino terminal on the cytoplasmic side. 2 hydrophobic pores contain the conserved asparagine proline alanine motif. Aquaporins selectively conduct water molecules in or out of the cell while preventing the passage of the ions and other solutes, and they are the integral membrane pore protein. So these are all about the aquaporins, and there is one thing which is mentioned here and I think we should remember is like the 2 hydrophobic, so 6 membranes spanning alpha this point and this point. So 6 membranes spanning alpha helical domain which both carboxylic amino acid terminal on cytoplasmic side is present there, and 2 hydrophobic loops contain conserved asparagine proline alanine motif. Okay. So, in the aquaporin there are two hydrophobic loop which contain the asparagine, proline, alanine motif. So, this question can be given to you like this, identify the conserved amino acid motif in the aquaporin. So, I think you should remember this part. Okay. So, I think it is clear what is aquaporin and what is it used for. I will move toward the next problem now. Okay. So, problem number 14, it says that which of the following statements is false regarding the retrovirus. Number A, it contains RNA as the genetic material. Number B, reverse transcriptase enzyme is present in the retrovirus. Number C, it proceeds through the DNA independent step. And number D, none of the above. So, you have to identify the false statement regarding the retrovirus. Read the options carefully and try to identify which might be the correct answer, means the false statement regarding the retrovirus. Number A, it contains RNA as a genetic material. Number B, reverse transcriptase enzyme is present. Number C, it proceeds through the DNA independent steps. Number D, none of the above. Okay, so do we have any correct answer for this? Yes, let me check. Okay, number D and number C and D. So, we have two answers actually, number C or number D. Now, we have to check if which is the correct answer. Okay, so number C is like it proceeds through the DNA independent step, and number D, it is none of the above. So, let us check which is the correct answer. C and D, right? Okay. Okay, so the correct answer is it proceeds through the DNA independent step, and this is the false regarding the retrovirus. Okay. So, it is the retrovirus, as you can see from this figure. So, it is the retrovirus. So, there is the envelope glycoprotein here, then we have this lipid envelope, then we have a protease enzyme, RNA matrix, the code, the reverse transcriptase, which is the enzyme present here, the integrase and the nucleocapsid. Okay. So, interesting points about retrovirus. Retroviruses, they contain RNA as a genetic material, but have DNA dependent steps in their replication. Okay. So, this point is very important. Retrovirus, they contain RNA as genetic material, but have DNA dependent steps in their replication. They replicate via transcription because the presence of the reverse transcriptase enzyme. Integrase, they transfers the viral DNA into the cell nucleus, and the viral double strand DNA is covalently and randomly integrated into the cell's genome. Retroviruses that can transform host cell at high rates contain gene sequences such as viral oncogenes and proto-oncogenes. Human retroviruses can cause immunodeficiencies, cancer and neurological disease. So, these are few important points about the retrovirus. Okay. So, now I will move toward the next problem. So, problem number 15, it says that which of the following is false regarding the renin-angiotensin system. Okay. Number A, aldosterone is a steroidal hormone. Number B, vasoconstriction is caused by the action of angiotensin 1. Number C, aldosterone enhances the retention of the sodium and water in the body. Number D, bradykinin is destroyed by angiotensin converting enzyme. So, this renin-angiotensin system, it is basically the system which is related to the blood circulation, and it is actually related to the blood pressure also. Okay. And how the blood is circulated and water and sodium or everything, it is related to this renin-angiotensin system. And just if you know, you can try to answer this question in the chat box, and which you think might be the correct answer, means this false statement regarding this renin-angiotensin system, and I will discuss this system in detail here, so you understand it. So, as I said, these topics are related to your week, your week 7 course content, but it is not directly mentioned in the previous year's question. So, I thought it is good to incorporate few different questions from the previous years, so that you understand the course content more better. And solving the assignments is not only the motto for your course. You should know understand the medicinal chemistry better. So, try to answer this question, read the question options carefully, and if you know you should want to try, you can try to answer this in the chat box. It does not matter if your answers are correct or wrong, but if you try, it is good. Okay. So, which of the following is the false statement regarding the renin-angiotensin system? So, do we have any answer for it? So, okay, good. We have one answer, number C. Others also try. Okay, let's check what is the correct answer or the false statement. Okay, so the false statement is the vasoconstriction is caused by the action of angiotensin 1. So, as I said, we will discuss this renin-angiotensin system in more detail. Okay, so renin-angiotensin aldosterone pathway. So, it is a very important pathway for our body, for the blood circulation. Okay, and there are some enzymes and some organs involved here, like liver is involved, the kidney is also involved here, and the lungs are also involved. So, what happens? Like the first step is the dehydration or sodium deficiency or hemorrhage. So, if it happens, like if you are dehydrated, you have sodium deficiency or hemorrhage, now what will happen? The decrease in the blood volume. Okay, so blood volume will be decreased, and the blood pressure is decreased due to this blood pressure is decreased because of this decrease of the blood volume. Now, what will happen if the blood pressure is decreased? The juxtaglomerular cells of the kidneys, they are activated. Okay, so in the kidney there are some cells which are called the juxtaglomerular cells, and if these cells are activated, when these cells are activated when the blood pressure is low, and then after activation of these cells, what happen? There is an increased renin. So, renin is a compound or a enzyme, you can this is a compound particularly. So, when the renin is increased in the body, and then what happened, the liver is also activated, and the liver is produced angiotensin, okay, and this angiotensin and this renin is converted to the angiotensin 1, okay. So, the angiotensin 1 will be increased. Now, what will happen if it is increased? So, this angiotensin 1, it is converted by the angiotensin converting enzyme or ACE in the and it is increased which is converted by this angiotensin converting enzyme to angiotensin 2. Now, what happened? This angiotensin 2, it will this increased angiotensin 2 will affect in 3 ways. First, the increased what will happen? Because of it, adrenal cortex is stimulated, and when the adrenal cortex is stimulated, what will happen? Increased aldosterone. So, aldosterone will be produced more, and if aldosterone is produced more in kidneys, increased sodium and water reabsorption. So, the kidney will reabsorb the sodium and water more because your body is feeling dehydrated. So, if the it is this dehydration is triggering the kidney this pathway, and now kidney will increase the sodium water reabsorption. Again, what will happen? Like what will happen, like this trigger of this increased sodium and water reabsorption, it will increase the blood volume, and when the blood volume is increased, what will happen? Blood pressure will increase until it is returned to normal. And all this is a one pathway, and another pathway, what will happen? If the angiotensin 2 is increased, the vasoconstriction of arterioles will happen and the blood pressure will increase to make it normal. So, this is the renin angiotensin aldosterone pathway. So, basically you are feeling dehydrated or your blood pressure is falling or you have a hemorrhage. What happens? Your blood volume is decreasing, resulting the lower blood pressure, and in this pathway what happen? Your blood pressure increases until it returns to normal. And there are this is the actually what happened with the person who are suffering from high blood pressure. So, this renin angiotensin aldosterone pathway, it is inhibited by the compounds which are your traditional I mean blood pressure controlling medicines. They actually inhibit this ACE enzyme and other enzymes involved in this pathway, so that the increase of blood pressure does not happen, okay. So, this is also something is like a high blood pressure. What happen? Like the sodium retention due to the aldosterone happens, and what will happen? The high blood pressure, and this high blood pressure why it happen? The oxidative stress damage to DNA vascular remodeling, okay. So, this is all about the blood pressure and the renin angiotensin pathway. So, number B, the vasoconstriction is caused by the action of angiotensin 1. It is the false, okay, because the this vasoconstriction, it is actually caused by the action of the angiotensin 2, not angiotensin 1, okay. So, this was all about your renin angiotensin system and the system by which how our blood pressure happens in our body, okay. So, now I will move toward the next problem. So, problem number 16, it says that which of the following natural product is used as antimalarial drug? Option A, Taxol, option B, artemisinin, option C, discoperol, and option D, curcumin. So, if you remember this question particularly, I actually discussed today in today's session only in first or second question, I think. So, try to answer this. I think you should be able to answer this. If you have listened the class carefully, you should have you must answer this question correctly, okay, because I discussed it. Which of the following natural product is used as antimalarial drug? Number A, Taxol, number B, artemisinin, number C, riscoperol, number D, curcumin. I expect more answers for this question because I discussed it today only. So, let me check. Okay, only one or two answer I think we get. No. Okay, so the absorption of sodium and potassium ion increase the blood volume. So it is actually basically okay. So this is for the last question. Okay, so I will come to this part first, and we have some answers. B and D are the most used for the next question. Okay. Okay, so your blood volume. So in the blood we have I think the blood volume, basically the blood, it is the water base. Okay, so this ions like sodium, potassium, these are very much I think related with the blood fluids. Okay, so the blood, you know, there are many components of the blood. Okay, it is not only the hemoglobin and all. So we have water, we have ions in the body fluid. Okay, so the increased sodium and as you say, as you have seen, I think carefully you should see here, like once I will highlight it. So there is in the kidney increased sodium and water reabsorption. Okay, so I have not said that only the increase of the sodium or potassium ion will increase the blood volume. There is water reabsorption also, and basically this water reabsorption is the main reason for the increase of the blood volume. But if you have dehydration, if you have deficiency of sodium ion, your blood pressure will also fall. Okay, so this in the kidney due to this aldosterone, not only the blood volume will increase, that will lead to the normal blood pressure, also it will cover up for the sodium deficiency, and sodium deficiency is also one reason for the fall of the blood pressure. Okay, so it may not be directly involved in the increasing the blood volume, but it is very much involved in the increasing the blood pressure. Okay, and okay, we have two options. So let us check what is the correct answer. So yeah, we have the correct answer, artemisinin. So few people have I think answered it correctly. Okay, so today I have discussed it in the previous one part. So this is the artemisinin. So what is artemisinin? Artemisinin is basically from a plant which is called the Artemisia annua. So it is a Chinese traditional medicine for the treatment of the malaria. Okay, so in the 2015, the person who have discovered this natural product, so it has been used in the traditional Chinese medicine from the years. In but one person, one professor, he identified this particular compound from that plant, from this lead compound, which is artemisinin, and it is used as a anti-malarial drug. Okay, so this is the structure of the artemisinin. As you can see, there is a tri-ring, three rings are joined together, and this is the artemisinin. So we have few derivative of the artemisinin. Okay, so one is dehydroartemisinin, which is basically here there will be a alcoholic group instead of the ketone. We have the artemether, which is we have this methyl group added with this alcohol if we have, and we have artesunate, which is this kind of derivative. So all these are the these are the derivative of the artemisinin, which is used for the malarial treatment. Okay, so what is the mechanism of action? So these compounds have the presence of endoperoxide bridge. So as you can see, this is the endoperoxide bridge which is present in the artemisinin. Okay, so this endoperoxide bridge interacts with the heme of the parasite. So as you know, malaria is it is caused by a parasite which is known as the plasmodium. Okay, so there are many plasmodium parasites like plasmodium falciparum, plasmodium vivax. Okay, so these parasites, they actually goes through our systematic blood circulation and mainly damage the liver. Okay, so and they basically damage the liver and the heme protein of the liver, but this endoperoxide bridge, they actually interact with the heme of the parasite. Okay, the heme iron cleaves this endoperoxide bridge, and there is a generation of highly reactive free radicals which damage the parasite membrane by covalently binding to the membrane protein. Okay, so what happened, like this artemisinin covalently bind to the membrane protein of the parasite membrane particularly, and then in that it goes to the heme protein of this parasite body, and this endoperoxide ring is cleaved, radicals are formed, and this radical will form the adapt with the heme protein and thus destroying the parasite. So artemisinins are actually the prodrugs of the biologically active metabolite dehydroartemisinin, which is active during the stage in which the parasite located inside the red blood cell. Although there is no conscious reading the mechanism through which artemisinin derivatives kill the parasite. Several lines of evidence indicate that artemisinins exert the antimalarial activity by the radical formation that depends on the endoperoxide bridge. When the parasite that causes malaria infects the red blood cell, it consumes hemoglobin within its digestive vacuole, a process that generates the oxidative stress. Okay, so it was all about the artemisinin, how it works and what it is. So remember, because it is a very important natural product. Okay. So now we will move toward the next problem. So problem number 17, it says that the following diagram represent the phenomena of SPR or surface plasmon resonance. So what is the represent here by the D? So as you can see, there is something is D here, okay, and it is what is this D stand for? Number A, monochromatic plane polarized light. Number B, flow of buffer solution. Number C, receptor. Number D, evanescence wave. So identify which is actually said by the D here: monochromatic plane polarized light, flow of buffer solution, receptor or evanescent wave. Try to answer this question in chat box. Try to answer what you think to be correct for this. So do we have any correct answer for this question? Yeah, okay, you understood this, which is good. Okay, so we have one answer, I think number D. Okay, let's check out. Okay, very good. So the correct answer is the evanescent wave. So let us see what is surface plasmon resonance and what is it all about. So yeah, if you look carefully to this thing, so this is the dextran layer. Okay, then there is a gold layer. So the dextran layer is for 100 nanomolar and 100 nanomolar and then nanometer, and then there is a gold layer which is about the 50 nanometer. Okay, then we have a glass plate, a detector, and this is the flow of the buffer solution. So the number C is the flow of buffer solution. Number D, it is the evanescent wave. Okay, number A, it is the immobilized ligand, and this is the monochromatic plane polarized light, which is the B. Okay, and then there is a detector. So what is surface plasmon resonance? So surface plasmon resonance is an optical method of detecting when a ligand binds to its target. The procedure is patented by Pharmacia Biosensor as Biocore and makes the use of the dextran coated gold surfaced glass chip. So there is a glass plate, after that we have a layer of gold about 50 nanometer, and then about 100 nanometer we have a dextran layer. This dextran is a polymer. Okay. So a ligand that is known to bind the target is immobilized by linking it to covalently to the dextran ligand, dextran matrix, which is in a flow of the buffer solution. Monochromatic plane polarized light is shown at an angle of incidence alpha from below of the glass plate and is reflected back at the interface between the dense glass coated glass and gold coated glass and the less dense buffer solution. Here a component of the light, which is called the evanescent wave, penetrates a distance of about one wavelength into the buffer or dextran matrix, normally all the light including the evanescent wave is reflected back, but if the gold flame is very thin, a fraction of the evanescent wavelength and the angle of incident is exactly right, evanescent wave interacts with the free oscillating electrons called plasmon of the metal flame. So, this is called the surface plasmon resonance. Okay. So, here the question was about this number D, which is called the evanescent wave. Okay. So, it is a component of light which penetrates a distance about 1 wavelength into the buffer or the dextran, and if the angle is exactly right for the incident plane polarized light and the gold flame is very thin, the evanescent wave interacts with the free oscillating electrons which are called the plasmons in the metal film. So, this is all about the surface plasmon resonance. So, now I will move toward the problem number 18. So, it is a math. Okay. So, when a medicine is added intravenously, its bioavailability is number A 50%, number B 25%, number C 100%, number D 98%. So, we discussed about the bioavailability in today's session only, in the previous one question, like in the question number 5 or 6 I think. So, in which we discussed what is bioavailability, and so use that knowledge to answer this question. So, when a drug is added intravenously means through injection directly to the system. Okay. So, what its bioavailability is? So, is it 50%, 25%, 100% or 98%? So, try to answer this in the chat box. What you think is the correct for this answer? Try to think about what is bioavailability, how we discussed it about the bioavailability. So, when a drug is given intravenously, what its bioavailability is? Is it 50%, is it 25%, is it 100% or is it 98%? Okay, let me check if there is any there are answers. Okay, number C. So, everybody is thinking it is number C 100%. Okay, let us check. So, yeah, very good. So, the correct answer is number C 100%. So, when a medicine is added intravenously, its bioavailability is 100%. So, in pharmacology, bioavailability, which is I think denoted by B, A or F. So, it is a subcategory of absorption and is the fraction or percentage of an administered drug that reaches the systematic circulation. By definition, when a medication is administered intravenously, the bioavailability is 100%. So, the fraction absorbed into the systematic circulation is the drug's bioavailability. So, as we have discussed, like the drug, it goes to the GI lumen, okay, the gastrointestinal system, then we have this gut wall, then portal vein, and this portal vein takes the drug to the liver. We discussed this just in today's session only. Okay. So, the bioavailability, it is equal to AUC. AUC means area under curve for the divided for the PO, okay, and then AOC by IV. IV means intravenous and PO means orally, okay. So, it is into 100%. So, it is a formula for the bioavailability. So, as you can see, there is a curve which is the bioavailability curve, and this is the when the drug is given oral, and it is when the drug is given intravenously. And the for example, this is the example which is given for this graph. Okay, so there is one more term which is called the bioequivalence. So bioequivalence is the term in pharmacokinetics used to assess the expected in vivo biological equivalence of the two proprietary preparation of a drug. If two products are said to be bioequivalent, it means they would have been expected to be in all intended purpose the same. Means if you are giving one drug in orally or intravenously, it will be equivalent because it will have same bioavailability. Okay, so this is all about the bioavailability. So in previously we discussed, like if you are taking a drug by your mouth, it will go to the stomach at first, then get dissolved, some part will not be dissolved, then it will go to the gut wall where it will be absorbed, some part will not be absorbed. So there are some loose for that, and then after from that gut wall by the portal vein it will go to the liver, and when it will reach to the systematic circulation. Okay, and the loose the drugs, the some components of the drug is getting lost, and the per component, the part which is completely reached to the fraction reached to the systematic circulation is called the bioavailability. So if you are giving a drug directly to the intravenous in the systematic circulation, means its bioavailability is 100%. Okay, now this related to the bioavailability, we have two maths today. So I will expect you to try to answer those mathematics. Okay, because the maths are very easy, so you have to just use this formula to solve the math. Okay, read the question carefully and try to solve. So the number 19, it says that if the bioavailability or F of the digoxin is 0.25 milligram tablet, in a 2.05 milligram tablet is 0.60 compared to the bioavailability of 0.75 in the elixir, 0.05 milligram per ml. Calculate the dose of the elixir equivalent to the tablet. Okay, so elixir means it is the injection form. Okay, so you have a drug which have for the bioavailability of 0.60 into 0.25 milligram tablet, and the bioavailability is 0.75 in a elixir which have 0.05 mg per ml. Okay, so 1 ml of that elixir has 0.05 mg of the medicine. So you have to calculate the dose of the elixir which is equivalent to the tablet. Number A 4 ml, number B 8 ml, number C 12 ml, and number D 25 ml. Take some time, try to take 2 to 3 minutes, try to solve this question. Okay, read the question carefully, it is very easy, but read the question carefully and try to answer it. If the bioavailability of digoxin is 0.25 mg, tablet is 0.60, to the bioavailability of the elixir is 0.75. When the elixir have 1 ml, in 1 ml elixir, now we have 0.05 mg of tablet. Calculate the dose of the elixir equivalent to the tablet. So do we have, okay, take 2 more minutes, then I will show you the answer. Try it for 1 to 2 minutes more. Okay, so did anybody have tried it. Okay, I think we have one answer which is number D. Okay, let's check which is the correct answer. Okay, so it is the number A. Okay, so I will show you how to solve it. See, it is very simple question actually. So, the drug bioavailable from the tablet is 0.25 mg. Okay, so the 0.25 mg have the bioavailability of 0.06, means 60%. So, what will be the 60% of this 0.025 mg? It is 0.15 mg. Okay. Now, if the bioavailability per milliliter per ml of the elixir is 0.05 mg into 0.075, means 0.0375 mg. Okay. So, if in 1 ml of the elixir have 0.05 mg of the drug. So, if you are given 1 ml, so what will happen? You will take 0.05 mg. So, if you are taking 0.05 mg, okay, so what will happen means, what it means like if the bioavailability is 0.75, means the drug which is reaching to your system is 0.05 mg into 0.075, means this much of mg. Okay. So, this much mg drug which is reaching to your system is in 1 ml of elixir. So, but to be equivalent to the tablet, you need this much of mg. So, this much of mg drug you will get from the XML, which is for 4 ml. Read the answer carefully and let me know if did you understand this or not. If you did not understand it, I will repeat it again, but read this carefully and tell me if I need to repeat it. So, do I need to repeat this? If the tablet's bioavailability is 0.60 means 60% and you are taking 0.25 mg tablet, means what is the amount of the drug which is reaching to your system? It is 0.025 into 0.60, means 0.15 mg. Now, the elixir means the liquid, it has 0.05 mg per ml. So, if you have if you are taking 1 ml, how much of the drug is reaching to your system? This is this much of mg. So, to be equivalent to the drug, you need this much of mg to in your system. So, if this mg it bioavailable from 1 ml of elixir, this mg is available from x ml, means x is 4 ml. Again, I am saying, if you did you understood it, understand this question or not? Do I need to repeat this math? Okay, nobody is saying anything, so I will think you have understood this. This is very easy actually. If you read the question carefully and then read the answer carefully, it is very easy. Okay, so we don't have much time to left today. So, I will quickly go to the last question. So, the problem number 20, it says that if the AUC or area under curve for an oral dose of the drug administered by the tablet is 4.5 mg per ml per hour and the intravenous dose is 11.2 mg per ml per hour, calculate the bioavailability of the oral dose of the drug. So, is it 30%, 40%, 15%, 55%? And I think I show you the formula, like what is the formula for the bioavailability? It is AUC of the oral divided by, I think, AUC of intravenous into 100%. Now, calculate it and tell me the answer. You can use this formula for okay, sorry. So, you can use this formula, right? The bioavailability is AUC of the oral divided by AUC of the intravenous into 100%. So, if you use this formula in this question, so what will be the correct answer? The AUC of the oral and the intravenous are given to you. Now, you have to divide and calculate it. Okay. So, did anybody try it? Okay, 40%. Okay, we have one answer. Okay, let's check out if the answer is correct. So yeah, very good, it is the 40%. So as I said, bioavailability, when the systematic availability of the drug administered orally is determined in comparison to its intravenous administration. So types of the standard use: intravenous dose is selected as standard, intramuscular dose for the poorly water soluble drugs, oral solution is used in certain cases. So the bioavailability of drug may be determined by the comparison of AUC data for the particular doses against the intravenous form. So if you have F is the AUC of dosage form divided by AUC by intravenous. So F will be 4.5. Okay, there is an mg and microgram, there is a unit difference here, so does not matter. So 4.5 divided by 11.2, it is 0.4 or 40%. So this is it. It was all about today. Now, so okay, so this was your week, I think week 6 live interaction session, when we discussed few things about the drug discovery process, things about the bioavailability, natural products. Okay, and I think please repeat question number 4. Okay, let's see which is the question number 4. Okay, so the question number 4, it was, there are several sources and methods for discovering new compounds. Which of the following is most likely to lead the discovery of complex structure quite unlike any other previously discovered? The options were combinatorial chemistry, database mining, screening plant extract, and Me Too drugs. So the plant extracts, screening the plant extracts are the most likely it will lead to the discovery of a complex structure which is not previously discovered, because as you know, the natural products, the structures of the natural products are very complex. So as I showed before, like the structure for the quinine, morphine and artemisinin, the structure of the natural products are very complex. So and only in the screening plant extract is the only method where complex structures will be discovered much faster than any other method. Okay, so this was about your problem number 4. So yeah, today it is all about the week 6 live session where we discussed your week 7 course content, and now I will close today's session, and if you have any doubt, anything regarding today's session, you can just drop an email to me. Okay, like every day I will share my email ID to you, and I have I think asked the NPTEL webs people to upload the once again. So I have asked them to upload the week this class materials, this lecture material, PPTs and the videos to the students, and I think they will do it shortly. Okay, so this was all about week 6, and for the next Saturday we will have the week 7 live session where we will discuss your week 8 course content. Okay. So if you have any doubt regarding today's session or any doubt regarding previous sessions also, you can drop an email to me, and now I will close the session, and thank you all for joining, and I will meet you in the next Saturday. So every Saturday we have this kind of meeting from 6 p.m. to 8 p.m. Okay, so let's meet to next Saturday from this time, and thank you all for joining today's session. I will now close today's session, and I will share this lecture PPT and today's session video shortly. Okay. So thank you all.